At a very young age, I became interested in the natural world. I've always been interested in specific types of plants that grow in very unique environments, but also how these very unique rocks and crystals are formed and where they come from. And that's led to my current research in cell biology, and specifically looking at how signaling functions in T cells, in your adaptive immune cells. My name is Katz. I'm a postdoc at the Salk Institute in the laboratory of Bjorn Lillemeyer. I'm a cell biologist, and I research signaling in T cells using single molecule microscopy. T cells are a very unique cell type. They're flowing through our bodies, they're cruising through, so they change their structure and form and their mobility often through our bodies. And they move very quickly. They react within seconds to less than seconds, and these reactions cause huge structural changes. These T cells are kind of your bouncers, your security system for your body. They identify whether the cells that are part of your body are sick or shouldn't be there, and they respond very quickly. Your adaptive immune system, which incorporates these T cells, are really important because when you don't have them, you're susceptible to all sorts of foreign invaders. And when you have them too active, you have autoimmune diseases. The focus of our research really is on the internal signaling mechanisms at the T cell receptor, where small molecules which would have better druggability within the human system could be targeted. The best way to visualize these things are with light microscopy, live cell light microscopy, and that allows us to see and visualize single molecules moving within these cells. And to better understand these cells, we can now think of how to manipulate them, how to treat them, and design drugs for future therapies. We have built a microscope that utilizes high energy lasers. These are lasers within the visual spectrum, and they illuminate molecules that we put into these cells that are tagged by what are called fluorescent proteins. That allows us to see these single molecules recruiting to the surface of the immune cell and moving. And that's actually allowed us to identify a new mechanism of how T cells react towards foreign invaders. It's now a key tool for identifying future drugs that we can use to change our immune response and allow our bodies to be more reactive towards foreign invaders, or to identify new drugs that could clear cancer cells from our bodies. I always had a lot of artistic materials around the house. I think that really ties into the creative and visual aspects of my research as well, using light microscopy to tweeze apart the finer workings of cell signaling and cell compartmentalization. I wouldn't be doing what I'm doing if I wasn't fundamentally interested in the science and it as a creative pursuit. I continue to use microscopes, I continue to collect plants, but it's become a profession.